All right. If anyone needs a seat, there's one here at the front and one here on the side. So if you want a seat at a table, then there are two available. So, all right. All right. It's so good to see you all. I felt sure after last week you wouldn't come back, but you're here, and that's awesome. So I get to guest myself today. So I'll give you a little bit of introduction as to why I'm actually standing in front of you now as a cancer researcher rather than one of your lecturers. And I'm going to tell you about a company that I started that's called Camaratech. And Camaratech, we presented at a very big medical conference in San Francisco in January. It's called the JP Morgan Healthcare Week. So it's a conference that goes for an entire week. About 50,000 people flood into San Francisco, all trying to show their science and do deals. And that's what we were there for as well. And one of the things that I had to make was a pamphlet, which, as you might remember, is one of your assessment items. This is my pamphlet. Doesn't look like a normal pamphlet, does it? I can pass these around. I've got 5 left over. But I actually had my business card stuck up in there and a little kangaroo pin, and they were really popular. So you've got to use everything you can to get your stuff noticed, and this is one thing. But as a consequence, I actually won a pitching competition, and we put Camaratech on the map a little bit. So only a little bit because we haven't got funding yet, but we're talking to a lot of people, and hopefully we'll go to the next step very soon. All right, now, my whole research project in Camaratech was founded on data that was generated by a PhD student. And I thought it was going to be worth introducing Mahanan to you to show you that this is possible for all of you, if this is what you want. So Mahanan came into my lab actually as a Wills student, so work integrated learning. And he did a project, wasn't associated with this one, but you know, he sort of got a taste for doing research, and there are certainly opportunities for you guys to get a taste for research still, because the VRES, Vacation Research Experience Scholarships, and there's an information session coming up very soon. If anyone's interested, 6 to 8 weeks full-time at the bench over Christmas usually, and it's a scholarship, so you get paid to do it. And you come in and you work with someone like me or one of the people that is going to be presenting to you, or someone who is in a different field if that's your cup of tea. And you come and you do research, but it's a scholarship, so it goes very nicely on your CV. And you get paid. It's only, I think, about $2,000, but it's $2,000, which, you know, will cover your rent for a couple of weeks. So it's well worth doing. So Mahanan came and did a work integrated learning type subject before that actually started, but then he did honours with me, and in between he went away and worked for a while, came back and realised he wanted to do science. Then he went away and travelled around South America and up into Alaska for 6 months, and then came back and started a PhD. And he finished his PhD just over 5 years ago. So we've actually had a very long research relationship and building what we do. And Mahanan is in fact a co-founder of Camaratech. So he and I started the company together. We got backing from one of Australia's largest venture capital firms to do it. It was worth a lot of money. And we achieved our milestones, but we're now looking for a new partner because they came for a short period of time, and that's what VCs do. They fund and they go away and then they come back, and we're talking to a new VC now. So hopefully that'll turn into something. But Mahanan's PhD was looking at a protein that is a receptor tyrosine kinase called FB4. And I've been working on FB4 for about 15 years. Sounds a bit tragic, but it kept me very, very busy for that 15 years. But Mahanan's project was to understand why FB4 is sumolated. So sumolation is a post-translational modification where a small sumo protein is covalently, so strongly, attached to a protein. And in this case, it's attached to the side chain, or the R group, of the lysine. It's reactive. So we wanted to know, why is it sumolated? What is it doing? And we discovered that it's sumolating FB4 and stabilising it, meaning FB4 hangs around longer in the cancer cell doing its job. And in the case of FB4, we know that it helps cancer cells move, so migrate. It also somewhat stimulates proliferation, but mostly it's a migration stimulus. So when Hannah was doing his project, we found this one member of actually quite a large family. There are 14 F receptors. And so there's another one that a lot of people are trying to target in cancer therapy development, and it's called FA2. So the next question was, well, are other members of the family also stimulated? And we found out that FA2 is as well. And then we looked even broader, because receptor tyrosine kinases, there are 58 different ones encoded by your genome. And a lot of them are very important in cancer, one of which I actually mentioned last week, and that's the epidermal growth factor receptor, EGFR. It also happens to be stimulated for stability. So the cool thing is, the lysine that is modified sits in a part of the folded protein in exactly the same spot. So we've identified a common mechanism, stimulation, on this particular lysine in many receptor tyrosine kinases. Not all of them. We've looked, not at all of them, but we've looked at some of the really important ones, and it's quite often there. And so our idea is that if we block this stimulation event, and we've chosen to do it using small molecules, so small organic molecules that we make or we take out of a library, then if we can block that, then that's a really fantastic new anti-cancer drug. Okay? Sounds pretty simple, right? Well, research isn't simple. But anyway, what we are doing falls into the category, and a brand new category of drugs that we call targeted protein degraders. So small molecules that cause a cancer cell to remove a protein it actually needs through a degradation process that is normal in the cell. But we take away this little flag, this sumoprotein, that stops that from happening, and instead the proteins are removed. So I'm going to show you what stimulation is so that you understand the process. But as I said, it's post-translational modification. And if we start here, here is the sumoprotein, and this is the C-terminal end. Now at this point, there's two different types, and that is because there are actually three different sumoproteins, two of which are identical, one of which is slightly different. But what happens with this sumo is it's made in a pre-form. So it's not ready to go yet. It's not functional. It has to be functionalised by a couple of things. And the first one is the C-terminal end gets cut off by a class of enzymes that we call CEMPs. So when that happens, we expose what's called a diglycine C-terminal end. Two glycines in a row. Glycine is very simple, it's an amino acid. But the sumo then gets picked up by this dimeric, so 2-unit enzyme called an E1 activating enzyme. SAE1 and 2 join together and they grab hold of this functionalised sumo and they give it a bit of energy. So they're adding ATP, and in doing so they build this new bond. So you can see that bond here between a cysteine that's in actually SAE1 and the end of the sumo. Then what happens is that uses the energy to pass it to this enzyme over here called an E2, E1, E2. This one is the only enzyme encoded in your genome that can do the next job. So there is only one of these. And it makes it a really lovely system to work with, because this enzyme, if we stop that from working, then we get no stimulation at all. There are some drugs that do that, and I'll talk about them later. But the E1 passes the sumo onto the E2, and once that's gone, the E2 goes off and finds target proteins that need to be modified. So it's coming through here, and here is our target that now has the sumo added. Now this can happen by itself. There are evidence that there are some proteins that don't need some help for the UBC9, but there are other ones that are called E3 ligases. And in this case, the E3 comes along and helps UBC attach it to the right lysine. So remember, our target proteins are a mixture of 20 different amino acids, right? So there's going to be a few lysines there. And the E3s help the UBC9 find the right lysine to actually modify. There are what we call consensus sequences, but we know that there are a lot of modification events that don't happen anywhere near a consensus, what they originally decided was a consensus sequence. So we don't know enough about how this works. But we do know the E3s are important. There's about 14 of those. But then there is one that has been described as an E4 ligase. And what it does is it forms chains. So SUMO2 and SUMO3 can actually form these little chains. SUMO1 probably can chain, but is mostly considered to be terminating. So it might be on the end of a chain. Or if it's attached somewhere on the protein, nothing else gets attached to it. So we have events where we have a single simulation event, but in a lot of proteins we might have more than one, maybe a couple. But there are lysine, a key lysine that's involved. Now once this happens and we've modified our protein, well, that can change a couple of things about how that protein behaves. It can change how it interacts with other proteins, so protein to protein interactions. It can change where the protein is in the cell. And in a lot of cases, it helps proteins move from the cytosol or cytoplasm across the nuclear membrane and into the nucleus. And an awful lot of proteins that are found in the nucleus are also simulatable. So we think it's a really important mechanism. And then the last one, of course, is the one that's interesting to us, and it can stabilise proteins. So these are the ones that we're trying to identify. Now, simulation itself is actually increased in cells to respond to stress. So if we heat you up a little bit, you will get more simulation, because if we heat you up, your proteins start to denature. And so we need to stabilise those. And you have a whole class of proteins called heat shock factors and heat shock proteins that come in, interact with the proteins, really key proteins, and keep them from being unwound and therefore not functioning. In the cancer cell, you probably don't realise this, but you don't have cancer cells, I have cancer, but cancer cells are actually really stressed. When they're growing in a body, they're proliferating out of control. They're pushing on each other. They're in mechanical stress. They're fighting for nutrients. They can't get oxygen. They can't get sugars. They have a higher metabolic requirement because they're growing so fast. And Jenny at the end is going to come in and tell you, actually I think she's sooner, she's going to come and tell you about the changes to metabolism that happens in cancer cells so that they can control, well actually they're very inefficient, but help with the energy production. But they also can't get rid of waste. So in respiration you convert the oxygen to carbon dioxide. Well, they can't get rid of it easily. So it builds up in the environment. And it actually contributes to making that environment around a cancer cell very acidic. That's actually something that people measure to try and identify where cancer cells might be. But all of these stresses mean that simulation is increased in cancer cells. And now a lot of proteins that should not be modified actually are. And these are really good things to find because they can make really nice and specific anti-cancer molecules. Okay, so our strategy from Camaratech is to identify proteins, we call them targets, that are specifically simulated in cancer cells and not in normal tissue. Because if we use small molecules to block that simulation event, hopefully, if it's involved in stability, it will instead cause that target protein to be degraded. And in that case, we have a new targeted protein degradation approach that could be applied to cancers. And we've chosen to apply it to triple negative breast cancer, TBNC. And the reason for that is TBNC is a fairly aggressive cancer that we cannot treat. Most of the breast cancers that women get, if they have estrogen receptor, progesterone receptor, and another RTK that we call HER2, then we can give them one of the drugs that target those proteins. Block the estrogen receptor. In hormone-dependent cancers like breast cancer, the early stages of those cancers actually need estrogen to continue to get the proliferative signal to grow. So we block the estrogens. With Herceptin, it's an antibody that targets and blocks HER2 which is related to EGFR, member of its 4-member family, and it works beautifully well for the individuals, the ladies, women and men, who actually make enough HER2 and whose cancers rely on that protein being produced. But it doesn't help everyone. Triple negative breast cancer, triple refers to the fact they don't make estrogen receptor, they don't make progesterone receptor, and they don't make any HER2. So none of the drugs that we have actually work for those people. So this is basically in a pictorial form what we're doing. We have a target that is simulated for stability, which enhances cancer cell growth, and we design small molecules to block this interaction to cause targeted protein degradation. Now, protein degraders are actually really popular at the moment as a new way of developing anti-cancer drugs in particular. And this is just to show you just how much money we're talking. The current global, so this in the world, TPD market is estimated at US 113, almost 114 billion US dollars, right? There is an awful lot of money being poured into this idea. Our idea is a little bit different though, because the ideas that they're currently spending money on are on molecules that look like this. Now, I know this is a little bit confusing and I'll explain it to you, but the current TPD technologies fall into really two classes. The ones that we call PROTACs, proteolysis targeting chimeras, that stands for, and there are similar ones that do different things, LITACs, and they come up with all these cool names, right? Or these little molecules over here that we call molecular glues. Now, has anyone heard of a drug called thalidomide? Heard of thalidomide? During the 60s, they used to give thalidomide to pregnant ladies as an anti-nausea. And unfortunately, it did other things in the developing fetus, and it caused problems with limb development in particular. So thalidomide is a molecular glue. So what a molecular glue does is it increases the interaction between a target protein and this little complex of proteins that we call an E3 complex. Now, the E3 complex is another modification called ubiquitin. So have you heard of ubiquitin? Well, SUMO stands for small ubiquitin-like modifier. So ubiquitin is the way that a lot of proteins are targeted or flagged for degradation. If we get a chain of ubiquitins like this on a target, it will be sent to a molecular machine called the proteasome. And the proteasome will chop up that protein and remove it. If we have a single ubiquitin, though, those proteins might get recycled and reused. So it's a chain of ubiquitin, polyubiquitination, that causes degradation. And this enzyme complex here is brought to the target through this hetero-bifunctional, hetero meaning different, bifunctional, two binding sites. So the molecular glue, or the much longer PROTAC, brings these close together, and now the E3 complex adds the ubiquitin to each of the target proteins, labelling it for degradation. Now, they've been working on PROTACs for about 20 years. The first one entered the clinic, FDA approved, so the US Federal Drug and Administration group approved the use for treating breast cancer. And they approved it, and what it targets is estrogen receptor. So for a start, it's only going to work in the ladies who actually have estrogen receptor, but it's being tested. The current idea is that it hasn't shown much improvement, which is a bit of a shame. And these molecules, these PROTACs, are actually really hard to make. These ones, we like them to be small molecules, meaning they have a molecular weight of under 500 Daltons. These guys are approximately or nearing 1,000, so 1 kilodalton. So they're a little bit big, because if you think about it, a lot of these molecules have to get inside the cell. They have to get past the plasma membrane, which you guys should remember from first year is a really good barrier. So it's quite challenging to get these sorts of molecules actually into the cell, which is why they started targeting things that are on the surface. But it's challenging. They're also really, really difficult to work with, and they're not always disease specific. But they show us something really, really important, and that is TPD technologies can work. Now, what we do, so much more simple. We still do targeted protein degradation, but we have a molecule that binds only a single site. So we just have to put it onto the target protein in the right place. We can be more disease specific because we're trying to block a disease specific modification. If the target and the E3 complex are in the same normal cell, then you're likely to stimulate degradation as well. And so there will be off-target effects, and more commonly with these sorts of drugs. For us, we're different to the traditional way. It's a new way of doing it. Oh, I thought there was another thing there. Anyway, so what we did was develop what we call the Camaratech platform. And a platform is just a process of how you're going to identify the targets, how you're going to confirm that the simulation, the modification, is actually disease specific, how you're going to identify the lysine that is modified, and then how you're going to build those drugs. So we have ways of doing all of this, and we've applied it successfully twice. I'll tell you all about what we call a demonstration target and show you how we did it in a minute. So we chose our favourite cancer target, EGFR, and in this case it's family member HER2, to demonstrate how our new technology worked. The whole point for this is new technologies people are already scared of. Put it in a new target as well, they're doubly scared. But show it works in something that everybody understands. And the biology of EGFR and HER2 is not entirely known, but is pretty well characterised, particularly in cancer cells. So we chose this from Mahannan's work as our demonstration of our ability. Well known, and good understanding of its biology, and it's a really validated target. So currently, EGFR is blocked with antibodies. It's on the surface of a lot of cells, so an antibody comes in and prevents it either from dimerizing or prevents it from binding to its ligand. Or inside the kinase function, the way it folds, we get small drugs to block the ATP binding pocket. So a kinase, hopefully you remember, I know Rachel really talks a lot about kinases. Kinases phosphorylate things. So they get the phosphate from ATP, but they have to bind ATP to collect the energy to then phosphorylate. And so a lot of these tyrosine kinase inhibitors actually block the ATP binding pocket. There are new ones that are coming out that are allosteric. Do you remember that word? Allosteric means it binds in another part of the protein but affects its conformation and therefore its function. Okay. The problem with a lot of these, though, is that tumours develop resistance. So there's a family of drugs, tyrosine kinase inhibitors, that has been produced by one of the biggest biotech pharmas in the world, and that's AstraZeneca. And AstraZeneca produced the very first inhibitor of this kinase. And unfortunately, after a couple of years, it worked beautifully well, and then it stopped working. People's tumours started to recur and come back. And it didn't matter. They tried to give them more drug. It didn't matter. They kept growing. And that is because the cancers mutate very quickly. It's sort of like selection of the fittest. Thanks, Darwin. So when we look at what is happening in the genome, mutations happen. We're not actually fixing the fact that mutations are occurring. We're fixing the consequences of those mutations. But in this case, with EGFR, you get specific mutations that mean the drug doesn't work anymore. And so what Astra has done, and they're almost now at their 5th generation, they make variants of those molecules. And the current one is called osimertinib. And it's starting to fail. So there'll be another one coming out soon. But they do work for a couple of years. And really, in doing that, and if you think about it, you're giving a patient another 2 years. And then giving them the next drug to give them another 2 years. And then hopefully giving them the next drug to get another couple of years. So you're extending someone's life and turning cancer from being an acute disease that will immediately kill someone, or within a couple of years, sometimes sooner, into a chronic disease that they'll have for the rest of their life, but is treatable. So that's probably the best we can hope for with a lot of cancer treatments at the moment. We're always hoping that we're going to find the magical target and the magical drug and everything will be great, but it's very, very difficult, as you will realise as we go through our course on cancer biology. All right, so the other thing about EGFR and HER2 is in the lab, and I'm sure you've heard of these sorts of experiments, you can do knockdown. So you can design little tiny RNA sequences like short hairpins or short interfering RNAs, and you pop them in a cell and they bind onto the message and they cause the message to be degraded before it's even made into a protein, right? When our Si or SH or whatever you choose, RNA sequences show when they knock them down that you actually need EGFR in a lot of cancer cells. So we know it's a valid target for removing the EGFR completely. So degradation, though, has not actually been achieved. There are plenty of people that are trying to. So they're making PROTACs, and one side of the PROTAC, remember they're heterobifunctional, one side is one of these small molecule drugs already. So lapatinib, gefitinib and afatinib are all variants of the original. I think gefitinib was probably the early one. And then they're attached via linkers to another thing that brings in that E3 complex. And the E3 complex is called either VHL or Cerebron. There's two different ones that people use. They're making the LITACs. LITAC just means it goes to the lysosome instead of the proteasome. And then there's lots of other molecules that they believe are actually degraders, including curcumin. You guys heard of curcumin? So you'll probably hear about it, you'll probably see some reading and stuff, but these are small molecules that are usually from other origins, other organisms like plants, for example, that are believed to have a sort of anti-proliferative effect. All right, now, the first thing that we had to do was prove that EGFR was actually simulated in our tumour tissue. So we did what's called a proximity ligation assay. You get a slide that has a section of a tissue on it, and the tissue obviously is a tumour tissue that you're interested in. And then, if inside those tumour cells you have your target protein simulated, and you add two different antibodies, the first one to SUMO and the second one to the target, and these are both primary antibodies, so they're recognising the original protein, then they'll bind. If you then look for both of those antibodies using a specific secondary antibody, now you guys have all done Westerns, right? You understand the process. You've done immunohistochemistry. This is fancy immunohistochemistry. Because what happens here is instead of having a fluorescent tag or some sort of enzyme, you have an oligo sequence, and these oligo sequences actually match. And when they match, they can then be the basis of a PCR reaction we call rolling circle amplification. So we add a polymerase, make lots and lots and lots of this circular bit of DNA that we've created because those two secondary antibodies are close enough together. And they have to be within 40 nanometres. So I know you know what a nano is. So they have to be basically next to each other, or in our case, attached to each other, to actually then be detected as a red signal. And that's what it looks like. So when we use a normal light microscope, we see all of these little red dots everywhere. And this shows us where our target and our SUMO are very close together. Yes, Katie?
What is the red protein that you've attached?
It's not a protein, actually. It's a little oligo probe that has a fluorescent or a red tag attached to it. So when it gets lots and lots and lots of it all attached together, you see it as a red signal in the one place like this.
And so you haven't stained any of the other tissue?
No. So when we use our PLA assay on, and in this case this is breast cancer, you can see this is tumour and this is tumour down here. The reason we know this is, can you see the nuclei? They're much, much bigger compared to the stromal cells that are sitting here. And I chose this one deliberately so I could show you the difference between what's happening here in the tumour versus the stroma, the normal cells in the microenvironment. And you can see there are virtually no dots here, and there are dots here. So we're definitely seeing that EGFR is simulated in the tumour cells and not the nearby normal cells. But we have actually looked at lots of different proteins, as you can imagine. We don't study just one. And this one here is what we call our commercial target. It's a secret. I'm not going to tell you what that is. I can tell you, but you'd have to sign a CDA, so a confidentiality agreement. But basically this one I can tell you is a transcription factor. Where do transcription factors work? In the nucleus. Where are the dots? In the nucleus. So you'll just have to trust me because I didn't put any normal cells up. This is cancer cell specific simulation of a transcription factor in the nucleus. And this is a fantastic target. So excited. Don't think I put it on there. This one, this is an oncogene. This is one of our very famous ones, and I'm not going to tell you which one either. There's only about 5. But this one gets mutated very easily. And if you have a mutated version of this protein, you will get cancer. So the problem with this protein is it looks like a very boring jelly bean. It has nothing. It's not almost smooth in terms of protein. So people have been trying to find drugs to stick in holes to stop it from working, but there are no holes. So currently this is an undrugged mutant oncoprotein. So we found out in pancreatic cancer, for example, there are red dots. So we know it is simulated. Trouble is we haven't quite worked out where yet. This one, this is my favourite PLA image. This is another undrugged, but now intracellular. So inside the cell, kinase. We have about 500 of those. But this one in particular is a really cool one that nobody else has actually drugged yet. And you can see just from the number of spots how highly relevant simulation is to this protein in pancreatic cancer, for example. So once we prove something is actually disease-specific simulation, we then have to work out whether it's important or not. So if we look at the process of simulation, all of these enzymes, enzymes are really good targets for drugs to stop the enzyme from working and then, determine what happens to the cell. So we have drugs like this one, TAK-981. It's from a company called Takeda. And what it does is it forms an adduct, which means it just sort of holds onto this complex. So once SUMO is there, it holds SUMO onto the SAE1 part of the dimeric protein and stops it being moved on. So it blocks the whole process right at this point. Now, Takeda are actually testing TAK-981 against cancer in the clinic at the moment. Problem is, it's blocking all simulation, and it's very good at it, but you have plenty of normal proteins that actually need to be simulated to have their normal function. So you're stopping all those from working too. And as a consequence, there are some pretty nasty side effects. So unfortunately for cancer patients, quite often we give them a drug like this because there's nothing else to give them. So it's very sad, but it's sort of proving that simulation is very, very useful and it's important to a cancer cell. Then there are other ways that we can do this in the lab. We can knock down UBC9. It's the only enzyme that does the next step. So if we remove it, we also stop processing or simulation of everything else. And then there is this really cool molecule called vermilion, which binds to the UBC9 enzyme and stops it from working as well. So this is what happens when we do 2 of those things. We use TAK all the time as well, but when we specifically target UBC9, we'll do this one first, so vermilion. You can see here, it's quite a high dose, 100 micromolar is very high, but when we add vermilion, we also lose EGFR. So this is just a Western blot, so we're looking at total protein here. Nothing's happening to our loading control, our GAPDH, and this DMSO, dimethyl sulfoxide, is an organic solvent that we have to use to make up our vermilion. So here, our vehicle, A, B and C are just triplicate repeats, and over here, A, B and C, with the vermilion we're losing the EGFR. So this would suggest that a consequence of treating cells with vermilion, which we believe to be blocking UBC9, causes EGFR to be removed. Okay, we don't know it for sure, right? This is just one experiment that we might do to test our theory. But over here, what we've done is we've knocked down using siRNA sequences, UBC9. And you can see as a consequence, we don't make very much UBC9 anymore when compared to a non-targeted siRNA control. So non-targeted, they're made of the same A, C, Gs and Ts, but they're just mixed up so that they don't actually target anything that we know about in the genome. So it's like a blank. And in this case, it doesn't do too much to the UBC9, and we get a good knockdown in the 3 replicates with the SI for UBC9. But look what happens to EGFR. It's completely gone. So it is very reliant on UBC9 being there. So our argument stands. We've tested our hypothesis again, and we think, well, okay, 2 different ways of knocking down UBC9 both have this effect on causing loss of EGFR. Right, now, what we have to do then is identify the lysine. And the way that we do this is using a process called site-directed mutagenesis, SDM. You guys heard of site-directed mutagenesis? So all we do in this is we clone the coding sequence of the part of the protein or the whole protein, depends on what we're doing. We clone it, right? The correct sequence, exactly what you guys are going to do in this prep class. And then we go in with primers and we create a version where we change the coding sequence of the lysine that we think is important to an arginine. So K to R mutation at one single point in the actual sequence. So when that protein gets made, it can't be sumoylated because sumo can't be attached to the arginine. And then we make these in the cells and we look to see what happens. So if that particular lysine was important to be attached to sumo to be stable, then when we take that away, we shouldn't see the protein. We should see that a message is being made, but the protein should be less stable, so we'd expect less protein. All right, so let's see what happens. So here we've overexpressed the normal sequence, wild type, and here we've made the mutant version where the K at a particular spot has been changed to an arginine. Now there's one more bit to this experiment because as you can see, we don't actually recover the protein. So how do we know we did the experiment right? We mightn't even manage to get our cells transfected and producing the protein at all. And we might be going, no, there's no protein there, but I've actually screwed up. So we use this drug called MG132 and it blocks the proteasome. Now we can only use it for a short amount of time, of course, because the proteasome does important things and removes unwanted proteins. But in the short time that we added, what we see is plenty of the protein. So we can rescue the protein by blocking degradation. So this proves to us that that lysine is sumoylated, and if we block it, change it, the protein gets degraded. So sumoylated for stability. All right, so then we have to do some fancy chemistry, and I'm not a fancy chemist. My COO, Chief Operating Officer, is a medicinal chemist, thank God. Very, very smart and lovely man, and he planned all of this bit. But basically, we use the fact that you can get crystal models for a lot of proteins, and EGFR is one of these proteins where we know pretty much exactly how it's folded, most of it. And when you look at the crystal model, you can work out what's, targetable, what has, little pockets that we can put a molecule in or have a molecule sit over the top. And we can try and target and stop not only simulation of the lysine by putting a molecule in the way, but remember UBC9, the enzyme has to bring the sumo to the target, so they interact. So we have a protein-protein interface that we could put a molecule in the way. So in this case, we do what's called in silico screening. And our in silico screening, computer-based screening, basically was done by a lady down at RMIT called Jess Holliam. And she wrote a program and curated a library of 6 million different drug-like compounds and took our protein and ran it through her screen. And she came back to us with about 315 molecules that she thought might be useful. So we bought them. And the first thing that we did was see whether they actually attached biophysically. So compound, to protein, see whether they actually interacted. And any that did, we kept. Any that didn't, we didn't work with anymore. Then we put them through a biochemical assay. Because simulation's a process, we make the enzymes, we make the sumo, we make the target, we mix it all together in the right buffer with a bit of energy, and we should be able to create a simulated target. If we add the compound, we should see less of that. And then finally, we tested anything that worked in both of these biophysical and biochemical assays against cells, because ultimately we're here to kill cancer cells. So we have to see how they behave when we treat them. So this is our model. This is the EGFR down here. We know there's a lysine here, this one, 722. So 722nd amino acid in the sequence of EGFR is a lysine that's modified. Up here is UBC9, and coming down through here is the end of the sumo, because UBC9 sort of grabs hold of the sumo. It has its little double glycine repeat hanging down. It comes over to a target, interacts, and it builds the bond, and then it disappears. Sumo's now attached. So what we did was recreate this bond because we knew it had to happen. And then we worked out what other amino acids were involved in holding that interaction together long enough. And believe me, 13 nanoseconds is long enough. But other interactions like ionic salt bridges, like some hydrogen bonds, and those sorts of things that actually allow this to stay together long enough to catalyse and build a new bond. This is a result of one of the assays that we do for biophysical. I'm not sure if you guys have heard of this, but it's SPR, surface plasmon resonance. If anyone's done a protein biochemistry subject, maybe with Dan Brozak? No? No? Anyway, what you do is you make your protein, you stick it onto a surface, usually it's a gold-plated chip, and then you wash your compound over, and if the compound binds, it changes how the resonance of the protein is on the chip, and that's measured. So a low concentration will give you a little signal, a big concentration will give you a much bigger signal, and from that we can work out an idea of how strongly they interact. We call that binding affinity, the KD. So we proved that some of our molecules bound to our target really, really well, and that was great because we're after those sorts of molecules. But we actually did a whole heap of other things as well, so native mass spec and nuclear magnetic resonance. I won't even begin to explain those because I have no idea. There are specialist people who know how to do this, and we pay a lot of money to them to do it. All right, so our biochemical assay, this is how it works. Make all of these proteins, add them together and get this. And that's what it looks like when we run it onto a gel and do maybe a Western analysis. So transfer it onto a membrane, look for it with an antibody and then a second antibody. And this is the modified band. Cool thing about proteins is, sumo's a protein, we're adding it to a target. As soon as we link them together, we've increased the molecular weight. So it shifts up. And as you can see, when we add our inhibitor molecule, we get less of that. The reason why we see any at all is because this is actually a one-way street. So once this bond is built, we don't break it because we're not adding the SEMPs, the enzymes that would break that. So it is a competition assay and we have to add a lot of drug to see it. This is when we were doing our screening, and one of the molecules we found was this one here, but these are all different molecules. And you can see, compared to the DMSO vehicle control, this is the only one that showed a reduction. And it turns out to be it's our favourite molecule at the moment. But here is our modified version. All of these things are actually the other proteins within the assay that also, for example, like UBC9, carry the sumo. UBC9 gets simulated too. So we see a lot of these things happening. And that's a good sign because we know then that we're not blocking the whole process. We're actually blocking that specific event. And that particular molecule is really nice. But we went from, in our second screen, from 638 compounds down to 72 that were shortlisted, and then 27 that we continued with, but 10436 is our favourite. We tested at a lower concentration and we saw it still did what we wanted it to. But how do we know that it's not doing things to other proteins? Well, we have to check. And there's a protein in our cells called RANGAP1 that acts as a protein involved in moving things from the cytoplasm to the nucleus. It's part of a nuclear pore complex. Does that make sense? And it sits there and it helps pass things into the nucleus using simulation. And so it's heavily simulated. And as you can see, when we add the TAC drug that Takeda made, blocks that first step, what we see is a reduction in how much this RANGAP1 protein there is. So we know TAC's working. TAC is really effective at blocking simulation of EGFR here and HER2 here. But here's our drug in 30, 9.5 and 3 micromolar. So we're using a dose response. So we'd expect more degradation here with a higher concentration and less down here. And you can actually, I'm hoping that you can see that. So there's much less here, much less of the HER2 compared to our DMSO control. So it is showing that we're targeting at least 2 members that we're hoping to, EGFR and HER2, and we're not affecting down here, here and here, the simulation of RANGAP1. But specificity is a really, really hard thing to prove when you're using a cancer drug. And proteins like tyrosine kinase inhibitors, for example, the tyrosine kinase is a particular domain within a whole heap of different proteins, over 500 of them, that all look the same when they fold. The subtle differences are in the positions of certain amino acids that makes them different to each other. But when we're trying to use something generic, even a tyrosine kinase inhibitor that's designed to target one will hit many, sometimes 20 or more. So you'll get non-specific effects. It's a common thing, and it's very, very hard to prove when someone says, this is the only protein my drug affects. Don't believe them. Seriously, they don't actually know. I don't actually know. I'm going to do all the tests that I can to make it as specific as possible, but I don't know. I can't look at all 26,000 proteins. Okay, so there's another way of measuring the interaction and the degradation. So this one is called a homogeneous time-resolved fluorescence, or HTRF assay. Here's our target protein, and this time we use 2 antibodies that recognise different parts of it, but are still close enough together that when you excite one of them with a certain wavelength of light, the fluorophore that's attached will emit light that excites the second one. So fluorescence resonance energy transfer from this one to this one, and when it gets excited, it emits light at a wavelength you pick up. Now, if you're going to pick up light here, it means these two are really close together and your protein's okay. But if we degrade it because we've added one of our Chimeric SIMO blockers, then those 2 antibodies will actually be far enough away that even if you excite this one, it's never going to be able to excite that one, so you'll never get a signal. So we use this assay to prove that we're losing the EGFR in the actual cells. And this is a key point. We have to show that this works inside a cell. It's not good enough to just be in an assay outside. Proteins fold, they interact with other things. They're in a biochemical environment. The pH has to be right, the salt has to be right. All of those things to fold the protein into its functional state. That's going to be in the actual cell. And it works. So here is our drug. Here you can see we've made a curve. You can see our degradation constant of 50% is 4.3 micromolar, which is actually pretty good. We want it to be under. We want it to be in the nanomolar region, but we're getting there. So, and we can target some of these either situations of cancers where there's lots and lots of copies of EGFR, where the gene itself is amplified in the genome, or the mutant versions that no one else can target yet. So we're doing really well, and you can see here that just to prove it, it's working nicely. Okay, so once we do that, we have to test them in lots of different cells. And as a demonstration target, EGFR is fantastic because its job is to cause cells to grow. activate proliferative pathways. So if we stop it from working, the cells just won't proliferate as much. And that works really nicely. This is what we see when we use a thing called an incucyte. Have you guys heard of an incucyte before? It's basically a cell culture machine that has a microscope inside. And it can actually look at every single individual well of a 96 well plate and do 6 worth of those plates in about 20 minutes. So it's a time-lapse image of your cells and how they're behaving. And after that, we can look and see whether one cell becomes 2 pretty quickly. And this is what happens when we just treat things with DMSO, these blue lines. Down here is when we use the highest concentration of the drugs. And you can see they're just not growing at all. So it's doing exactly what it should. It's degrading EGFR and stopping the cells from proliferating. But the weird thing happened. So this is how a normal proliferation assay would look. So this is in DMSO. And you can see the cells are sitting down, spreading out. When they go to divide, they round up, they split in 2 and they sit down. And over a period of time, we get more and more cells, right? We'll stop that one because I want to show you what happens when we add one of my favourite compounds here, but in a concentration that is too high. And when I say too high, watch what's going to happen in these boxes up here, and even this one in particular. So let's run it. So we're getting to up to about 8 hours. Can you see there's something happening over here, 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 here? You see there's a little brick forming? That's our drug coming out of solution and forming a crystal. So it's not actually getting in the cells to target the protein, is it? It's falling out. So this is too high a concentration. And you know what? Cells don't care when it's a crystal, because look at this one. It's growing right on top of it. So that was fairly disappointing, but it means that this drug could never be a drug. So we have to do some fancy chemistry to make it more drug-like. The good thing is they're not actually toxic, because that's another thing. When you actually kill a cancer cell, you want to activate an apoptotic pathway. You don't want it to be toxic, because if it is, it'll pop everything open, all of the cell contents will leak out, and it'll damage the tissues around it. You want it to be controlled so that your immune cells, your phagocytes, can come in and clean up the mess, right? Recycle what they need. So we tested this in 2 different cells. These ones, heck, look at the time. And these ones, which is, I think, I can't remember, lung cancer maybe. And you can see 10436 only starts to see an effect here, but that's because we're actually killing the cells, which is great. And this one over here doesn't do anything to normal human embryonic kidney cells, so that's awesome. But we have over 400 targets, and guess what? I put 40 of them into a list for you to work with. So your project is to choose the gene, and a lot of you have done this, not everyone, so chop, chop. Choose the gene, write me a paragraph, and this is your hypothesis for your study, right? And we're going to make a construct where your protein is created with a green fluorescent tag, and then we're going to look and see where they are. And in this case, this is where SUMO goes in a stressed cancer cell. It goes into these little spots that we call nuclear stress bodies. And that's it. So sorry I got right up to the hour then. I can stay here and ask and answer any questions from anyone who wants to. Anyone who has a question and they don't want to fill out the sheet can email that question to me. But yeah, I did sort of take too much, rabbit on too much, so that's on me. All right.